I want you to picture the last time you walked through a city park.
Yeah.
Or, maybe just that little patch of grass between the sidewalk and the street.
Mm-hmm.
You're probably looking at the green, maybe trying to dodge a muddy patch. But for the most part,
Yeah.
you're ignoring the details.
Oh, absolutely. It's the background noise of nature, isn't it? We have to filter it out. I mean, if we paid attention to every single blade of grass, we'd never get anywhere.
Exactly. But if you actually stop for a second and look down, I mean, really look, you'll see them. They are everywhere.
They are.
Small,
Yeah.
brown, and completely unremarkable at first glance. In the birding world, they have this term for confusing little birds, LBJs, little brown jobs.
Right.
Well, in the fungal kingdom, we have a very similar and maybe a slightly dismissive acronym, LBMs.
Little brown mushrooms. And they are the bane of every amateur forager's existence. I think that's fair to say.
It is. And the standard advice you always hear for LBMs is, well, it's pretty blunt.
Ignore them.
Just ignore them. They're too small, they're way too hard to identify, and frankly, the risk of mixing them up with something deadly is it's too high.
Yeah.
They are the do not touch section of the forest floor.
Which is a shame, at least scientifically speaking, because by ignoring all of them, we're ignoring one of the most successful biological conquests in recent history.
And that is exactly why we are here today. We are not stepping over the LBMs. We are going to get down on our hands and knees right into the wet grass to look at one specific, incredibly common and deceptively complex mushroom,
Agrocybe Pediades.
the common fieldcap.
And common is absolutely the operative word here. You could argue it's one of the most widespread mushrooms in the human-altered world. Lawns, parks, wood chips, it's everywhere.
I was just reading through our source stack for this, which, by the way, is a wild ride today.
It really is.
We got field guides from Connecticut, from California. We have these intense phylogenetic studies from Russia, ecological surveys from the Brazilian savanna, and some pretty terrifying toxicology reports involving dogs.
Yeah, that part is. It's sobering.
It is.
Yeah.
And the picture that emerges from all this isn't of a boring little mushroom. It's a picture of a master of disguise.
It really is. Agrocybe Pediades is sort of a masterclass in mycology because it highlights everything that makes studying fungi so difficult and so interesting.
How so?
Well, it changes its appearance based on the weather. It has a genetic footprint that literally spans continents. And it has this identity crisis that has forced scientists to completely rewrite the textbooks.
So the mission for this deep dive is to take this invisible neighbor, this thing we walk past every single day, and to decode it. We're going to look at its biology, its global travel itinerary, and the molecular detective story that proves it's all one big, happy, chaotic family.
A very chaotic family. And we should probably start with where you actually find it because, you don't need to hike deep into the wilderness for this one.
No, you really don't. This is the mushroom of the Anthropocene. It loves us. Or maybe it's more accurate to say it loves the messes we make.
That's a very fair way to put it. Agrocybe Pediades is a saprobe.
Okay, so break that down, a saprobe.
It just means it's a decomposer. It feeds on dead organic matter. But unlike some really picky mushrooms that need, say, a specific type of ancient rotting oak tree, Pediades is not picky at all.
It's a generalist.
A huge generalist. It loves grasso, lawns, meadows, pastures. It loves dung. And it absolutely adores wood chips.
Ah, the wood chip mulch connection. That's so interesting. You see these huge piles of mulch in suburban landscaping, and then a few days after a good rain.
They're just covered in these little brown caps. It's prime habitat. It's taking advantage of this incredibly nutrient-rich environment that we've just created for it and spread everywhere.
And what about seasonality? When should you be looking for it?
You're mostly looking at late spring into early summer. That's its peak, which is one reason it often gets confused with the spring fieldcap, which is a different species, Agrocybe precox. Right. But in warmer climates, or, even just during a warm, wet spell in the autumn, it can pop up pretty much whenever there's enough moisture.
So it's very opportunistic.
Highly opportunistic, and that opportunism is reflected in its appearance. You mentioned it's a master of disguise.
I did.
That starts with the cap itself.
Okay, so let's break down the morphology. If I'm kneeling there in the grass and I'm looking at a cluster of these, what am I actually seeing? Because the descriptions I read in the guides seem to vary wildly depending on how old the mushroom is.
They do, and that is the very first trap that people fall into. When Agrocybe Pediades is young, the cap is small, usually what, 1 to 3 centimeters, maybe up to 3.5 if it's a real monster.
Niche.
Very small. And it starts out hemispherical.
Like a little dinner roll or a tiny bun just sitting there on the ground.
Exactly, a little convex bun. But
Yeah.
as it matures, that cap expands and it flattens out. By the time it's fully mature, it can be almost completely flat.
Okay, so the shape changes. But the real trick, the thing that really throws people off, is the color change, right? There's a technical term for it.
There is. The term is Hygrophanous.
I love that word, Hygrophanous. It sounds like a superpower, but it's really just about water retention, isn't it?
Essentially, yes. It just refers to the tissue's ability to change color as it loses or gains moisture.
So what does that look like in practice?
When Agrocybe Pediades is fresh and wet, say, right after a morning rain, it's this lovely, sticky, tawny brown color. Think dark honey or a deep caramel.
Okay, so rich, warm, brownish yellow.
Right, a very saturated color. But as the sun hits it and the wind starts to dry it out, the color just, it fades. It fades rapidly.
Rapidly.
Within hours, it'll go from that tawny brown to a pale brownish yellow, then to buff, and eventually it can end up an off-white or almost a cream color.
So if I see a dark brown mushroom sitting right next to a cream-colored mushroom in the very same patch of grass, I might just assume they're two totally different species.
And many people do. Early mycologists did too. That's where a lot of the confusing old names came from. But it's the exact same mushroom, just at different levels of hydration.
It's like seeing a wet t-shirt versus a dry one. Same shirt, totally different color value.
That's a perfect analogy for it.
There's another feature I read about regarding the texture that I found really distinctive. The notes mention that as it dries, the cap doesn't just fade, it actually changes its structure.
Ah, you're talking about the cracking.
The cracking, yeah.
This is a classic field mark, though it's not totally unique to this species. As the mushroom expands and the sun bakes it, the skin on the cap, the pilipellis we call it, it can start to fissure, it can crack.
And what does that look like?
It ends up looking like a dried up riverbed or cracked mud. You get this network of little fissures across the surface.
I saw there's a related species, Agrocybe dura, where this is basically its defining feature, right?
Yes, Dura is often called the cracked earth field cap because it does this so dramatically. But our mushroom, Pediades, does it too, especially when it's growing out in exposed areas like lawns.
It gives it this kind of weathered, rugged look, which is pretty funny when you think about how small and fragile it actually is.
It really is.
Speaking of fragile, we have to talk about the veil. This seems to be the part of the mushroom that causes the most confusion for people trying to identify it.
The case of the vanishing veil. It's almost poetic, biologically speaking.
So just to set the stage, what is a partial veil in the first place?
Okay, so in the mushroom world, many species have what's called a partial veil. It's a protective membrane that covers the gills when the mushroom is just a baby, a little button. It keeps the developing spores safe and moist until they're ready to drop.
It's like the safety seal on a jar of mayonnaise.
A biological safety seal, exactly. Now, in many mushrooms, when the cap expands, that seal breaks. And usually, it leaves evidence behind.
Like what?
Well, it might leave a ring around the stem, that's called an annulus, or it might leave little fleeky patches on the cap. Think of the ring on a portobello mushroom from the store, or the classic skirt on a deadly amanita.
But Agrocybe Pediades is different.
Very different. Agrocybe Pediades has a veil, but it is incredibly ephemeral. It is so thin, so wispy, and so fleeting.
How fleeting are we talking?
Well, the source from mushroomexpert.com, Half-Dash, which is a fantastic resource, by the way, notes, that you practically have to be looking at buttons the size of pencil erasers to even catch a glimpse of it.
So if I sleep in until 10 in the morning, I've probably missed the whole show.
You have almost certainly missed the show. It leaves these tiny, cottony, white remnants on the very edge of the cap margin. But as that mushroom grows, those remnants just dry up and blow away, or they just disintegrate within hours.
And crucially, this is the key point. It does not leave a ring on the stem.
Correct. No ring. And this is a huge diagnostic point for identification. There are other common Agrocybe species, like that Agrocybe precox we mentioned, that typically do have a persistent ring.
So people find Pediades, they see that it has no ring, and they get confused because it looks like an Agrocybe in every other way.
Exactly. They don't realize that the absence of the ring is the feature. It's a ringless field cap.
It's defined by what's missing. That's very zen.
It is. And, it complicates things because you have to trust that the ring wasn't there to begin with, rather than assuming it was there and it just fell off.
Right. Okay, let's look under the hood, or I guess under the cap. The gills. What's the story there?
The gills tell us a story about the mushroom's life cycle. When the mushroom is very young, the gills are whitish, maybe a pale gray or a cream color.
And how are they attached to the stem?
They're adnate, which means they're attached broadly to the stem, or sometimes adnext, which is a narrower attachment. But the big story with the gills is the color progression as the spores mature.
It's a dramatic shift, isn't it?
It's a huge shift. They go from that pale cream color to a dull, mature brown.
The notes I read describe this color as cigar brown.
That's the classic description, and it's a very specific brown. It's not a rusty orange brown like a deadly Galerina. It's not a purple-black like a Stropharia or a Penelazes. It's a dull, earthy tobacco brown.
And that color is coming directly from the millions of spores that are coating the gills, right? So if we were to do a spore print, and for anyone listening, that's when you cut the cap off a mature mushroom and place it gill side down on a piece of white paper or foil overnight, what would we see in the morning?
You would see a dark brown print. Sometimes you might see a hint of cinnamon in it, but it's predominantly a dull, dark brown. And that spore print color is the #1 way mycologists separate the entire genus Agrocybe from other lookalikes you find in the grass.
Okay, so if you get a rusty print,
Drop it immediately. That's a huge red flag.
and if you get a purple-black print,
It's something else entirely, probably a Penarlis or Stropharia. You are looking for that specific cigar brown.
before we leave the macroscopic description, the things we can see with our eyes, we have to mention the smell. I found this part funny because describing smells is so subjective, but the literature is very, very consistent on this one,
It is surprisingly consistent. The term everyone uses is farinaceous.
which is just a very fancy way of saying mealy.
Yes, it smells like raw flour or wet grain or maybe bread dough that hasn't been baked yet.
I always try to imagine what that smells like out in the field. It's not that mushroomy smell you get from a grocery store button mushroom. It's more earthy and almost bland.
It's a distinct, slightly yeasty, grain-like odor. And while it might not sound very exciting, it is a critical identification feature. Many other little brown lawn mushrooms have no smell at all, or they might smell like radishes or something vaguely chemical.
So if you crush a little piece of the cap between your fingers and you get that whiff of raw pastry, you're on the right track for Agrocybe.
You're definitely in the right ballpark.
Okay, so we've looked at it, we've smelled it, we've watched it change color in the sun. But as we established right at the beginning, LBMs are tricky. The visual inspection, the macro features, that only gets you so far.
It really does.
To really understand what Agrocybe Pediades is, we have to zoom in. Way in, we need the microscope.
This is where the detective work gets really serious. Because to the naked eye, a small brown mushroom is, well, just a small brown mushroom. But at 400 or 1,000 times magnification, Agrocybe Pediades reveals its unique machinery.
Let's start with those spores. We know they're brown in mass from the spore print, but what do they look like individually under the scope?
Individually, they are ellipsoid to ovoid, so basically egg-shaped. They have quite thick walls, which makes them durable, helps them survive. But the real standout feature is what's called the germ pore.
I saw this mentioned in all the technical descriptions. A germ pore. Is that what it sounds like? A hole for the germ to come out of?
That's basically it, yes. It's a specialized area, usually at one end of the spore, where the cell wall is either thinner or modified in some way. Under the microscope, it looks like a distinct pore or a kind of flattened, truncated spot at the apex of the spore.
And in Agrocybe Pediades, this pore is pretty obvious,
It's quite prominent, yes, up to 2 micrometers wide.
which is obviously incredibly tiny, but in the microscopic world, that's a gaping hole.
It is a significant feature,
Yeah.
and its function is vital. When that spore lands in a suitable environment, say a nice pile of wet wood chips, that pore is the escape hatch. It's where the germ tube emerges to form the new primary mycelium. Without that pore, the genetic material is essentially locked inside the fortress of that thick spore wall.
It's the door to the next generation.
Precisely. And not all mushroom spores have them, so seeing a large, distinct germ pore on a thick-walled, smooth brown spore is a huge, huge clue pointing you toward the genus Agrocybe.
Okay, that's the spores. But then we have these other microscopic structures. The sterile cells, the cystidia. I always think of these as the unique fingerprints of a species.
They really are. Cystidia are fascinating. They are cells on the gills that don't produce spores. They're sterile. They might be structural or protective, or maybe they help with evaporation. We're still debating their exact function in some cases.
But their shape is what's important for ID.
Their shape is taxonomically vital. In Pediades, we're most interested in the Chylocystidia.
Chylocystidia. So those are the ones on the edge of the gills.
Exactly. Chylo for lip or edge. And the shape is described as ligeniform.
Ligeniform, what does that mean?
It means flask-shaped. If you can picture an Erlenmeyer flask from a chemistry class, it has a wide, swollen base that tapers into a narrower neck.
Okay, I can see that.
And sometimes the neck of that flask has a little knob on top, which we call capitate. So it's a flask with a little head on it.
A flask with a head. Got it.
And just as important as what is there is what isn't there.
Right.
Agrocybe Pediades typically lacks pleurocystidia. Those are the sterile cells on the face of the gills. Some other look-alike Agrocybe species are covered in them. So if you look at the face of the gill under the scope and it's just a sea of spore-bearing cells, the basidia with no weird flask-shaped monster sticking out.
That's another point for Pediades.
Another check in the box.
It's just amazing to me that we have to go to that level of detail, counting microscopic flasks, just to confidently identify something that grows on pretty much every lawn in America.
That's the joy and the madness of mycology. The macro is deceptive, the micro is definitive.
Well, usually definitive. But as I was reading, even the micro features have led to some serious arguments over the years. And this brings us to what I think is the real intellectual meat of this deep dive, the genetics. You have this study here from Russia, Malasheva, and Kiyashko that really blew the lid off the classification of this mushroom.
This is a landmark study for this particular group of fungi. It directly addresses the classic lumper versus splitter debate.
Before we dive into the data, can you frame that debate for us? Because this happens in every branch of biology, doesn't it, this argument?
It absolutely does. Splitters are the scientists who look at any variation and they see a new species. They want to give everything its own unique name.
So a splitter would walk through a field.
And they'd see an agrocybe that's a little bit paler and it's growing in sand. They'd say, aha, this is Agrocybe ranicola, the sand-loving wort.
Okay.
Then they'd see one a few feet away that's perfectly round on top, and they'd say, and this must be Agrocybe semiorbicularis. They want these precise names for every single variation they can see.
And the lumpers, what's their take?
The lumpers look at the big picture. They say, no, wait a minute. These are all the same organism. It's just short because it's growing in a cold spot, or it's pale because the sun dried it out.
So they argue for something called phenotypic plasticity?
Exactly. The ability of one single genotype, one set of genes, to produce a bunch of different physical forms or phenotypes based on the environment it's in.
So, historically, Agrocybe Pediades was what splitters had a field day with. It was a mess of different names.
A total mess. In the older field guides, you had A. Pediades, A. arenicola, A. Sapediades, A. semiorbicularis, all treated as separate, independent species.
And then enter the Russian team and their DNA sequencers. What did they actually do?
They took a molecular approach. They bypassed the physical appearance and went straight to the genetic code. They analyzed what's called the NRITS region of the DNA.
The ITS region, I've seen that mentioned a lot.
Yeah.
That's the standard fungal barcode, right?
It is. It stands for internal transcribed spacer. It's a section of non-coding DNA in the ribosome genes that mutates at a rate that is just right for distinguishing species.
What do you mean, just right?
It evolves fast enough that different species have different sequences, so you can tell them apart, but it evolves slowly enough that all the members of the same species, and even the same genus, share a lot of similarity. It's the gold standard for this kind of work.
And they didn't just sample mushrooms from one backyard in Moscow.
No, this was a massive trans-Eurasian effort. They sequenced specimens from St. Petersburg in the west, all the way across Russia to the Far East, and down into Central Asia.
And they were taking specimens that had been identified morphologically as all those different species.
That's the key. They took the sand ones, the round ones, the ones with a little bulb on the stem, all of them, and they sequenced them.
The verdict, lumper or splitter?
The verdict was a complete, total landslide victory for the lumpers.
They were all the same mushroom.
Genetically, they formed one large, very well-supported clade, a single branch on the evolutionary tree. There was practically zero genetic distance between a specimen that was labeled arenicola growing in sand and a specimen labeled Pediades growing in rich soil.
So the DNA just said, I am all one thing.
That's exactly what it said.
That is profound.
Yeah.
It means that all of those physical differences, the things we spend hours describing in field guides, the cap shape, the color, the texture, they're just superficial wardrobe changes.
Exactly. It confirms that Agrocybe Pediades is an incredibly variable species. It has a very high degree of polymorphism. It can look like a dozen different things depending on where it puts its roots down, so to speak.
But there's another layer to this study that I found even more fascinating. It's the geographic uniformity.
Yes.
Usually, if you have a population of animals, say, in St. Petersburg and another population in the Far East, thousands of miles away, they start to drift apart genetically over time. You get distinct subspecies. But these mushrooms didn't. Why not? Yeah.
This is where that Anthropocene angle comes right back in. The authors suggest an anthropochorous distribution.
Okay, you have to break that word down for us. Anthropochorous.
Anthropos meaning human, and chorus relating to dispersal or movement. It means that we are the vector, humans have spread it.
We are the wind.
We are the wind, we are the river, and we are the migrating bird, all rolled into one. Think about it. We move massive amounts of soil for construction and landscaping. We ship turf and grass seed across continents.
And animals, too?
We move cattle and horses, which carry spores in their digestive tracts and on their hooves. We move hay and straw and wood chips for our gardens.
So what you're saying is, we have been mixing the gene pool of this mushroom so aggressively and so constantly that the populations have never had a chance to become isolated and diverge genetically.
That's the theory, and it's a very strong one. A mushroom in a lawn in California might be genetically identical to one in a lawn in Moscow because they both hitchhiked on the same global agricultural supply chain.
We've created a cosmopolitan species. It's not just a natural occurrence. This mushroom is a companion to human civilization.
It is. We are its primary dispersal agent now.
That blows my mind.
Yeah.
We think of nature as this separate thing over there. But in the case of the common fieldcap, we are the bus driver.
We are. However, I have to add one little nuance here. The study was a huge win for the lumpers, but the splitters won one tiny battle.
Of course. There's always an outlier, isn't there?
There always is. They found one small subclade, a little group of specimens within the main group, that was genetically distinct enough to deserve a separate name. They decided to call it Agrocybe Pediades variety Bispora.
Bispora. I'm going to guess that has something to do with the spore count.
You got it. Most of the mushrooms in the Agrocybe Pediades complex have 4 spored basidia. Those are the little cells on the gills, and they produce 4 spores each. This variety consistently produces only 2 spores per basidium.
And were there any physical traits that went along with that?
Yes. Morphologically, it tends to have a little bulbous base on the stem, a little swollen foot at the bottom.
So it's kind of a combination of the old concept of Agrocybe Pediades, which was known for the bulbous base, and this 2-spored feature.
Exactly. So the authors proposed keeping it, but just as a distinct variety. Not a full separate species, but Agrocybe Pediades var. Bispora. It's a nice little nod to the fact that evolution is still happening. Nature's still trying to create variation where it can.
So we've covered its genetics across Russia and Asia. But to really prove this global traveler theory, we need to go somewhere completely different. Let's talk about Brazil.
Yes, this brings us to the Kalasa et al. Paper from 2020. And this takes us from the cold Russian steppes to the tropical savanna.
The Cerrado.
The Cerrado, which is this fascinating biome. It's a vast tropical savanna with really distinct wet and dry seasons. It's hot, it's intense, and historically, it's not the kind of place you would expect to find a temperate lawn mushroom like Agrocybe.
So before this 2020 paper, had anyone ever seen an Agrocybe there?
There were no formal records of the entire genus in the Cerrado biome. Plenty of other amazing tropical fungi, for sure, but not our little fieldcap.
And then they found it. But they didn't find it deep in some untouched nature reserve, did they?
No, and this fits perfectly with our anthropochorous theory. They found it in the state of Goiás, in an organic vegetable garden, and specifically growing directly on cattle dung.
Ah, there it is again, the cow connection.
The cow connection is absolutely key. This confirms the fimicalist nature of the species in this region. Fimicalist just means dung-loving.
Dung-loving feels like a very brave lifestyle choice.
For a fungus, it's a brilliant choice. Dung is packed with nutrients, nitrogen, undigested plant fibers. It's a 5-star buffet.
But the significance of this find isn't just that it likes poop, it's the climatic adaptability, right?
That's the real story. Russia is cold, the Cerrado is hot, and it has a brutal dry season that can last for months.
And this mushroom has to be able to survive that. Scorching heat, long periods with no rain.
Exactly. The fact that it popped up there shows an immense physiological plasticity. The researchers were so interested, they actually took it back to the lab to see how it behaved in culture.
And what happened in the Petri dish?
They grew it on something called potato dextrose agar, which is pretty standard fungal food.
Yeah.
It produced this lovely white, downy mycelium, but it grew pretty slowly.
That's slow.
About 7 to 8 centimeters in 4 weeks, which, some molds can cover a plate in a day or two.
So it's not aggressive.
It's not aggressive, but slow and steady wins the race when you are built to be stress tolerant. And microscopically, the culture showed hyphae with clamp connections. These are little bridges between fungal cells that are characteristic of this group of fungi, the Basidiomycetes, which helped confirm the ID.
So being able to culture it suggests that this strain has really adapted to those local conditions.
It does. We now have a mushroom that can handle the Russian winter and the Brazilian summer as long as humans are nearby with our cows and our gardens and our wood chips.
He is the ultimate generalist. It follows the plow and it follows the cow.
That's a great way to put it.
Now, we've been calling it Agrocybe Pediades this whole time. And clearly the Russian study confirmed that Pediades is the correct name for this core group. But I've heard rumors of a breakup in the genus, something about a new name, Cyclocybe.
Ah, yes. The great taxonomy shakeup of the 2010s. This is a classic example of playfully ruining everyone's nice, orderly lists.
You have to explain polyphily for us.
Okay, so a polyphyletic group is when a group of organisms are all put together in the same bucket because they look alike, but they aren't actually closely related evolutionarily.
So it's like putting dolphins and sharks in the same family, just because they both have fins and swim in the ocean.
That is the perfect analogy. They evolved those features separately. It's convergent evolution.
So the genus Agrocybe, as we used to know it, contained both dolphins and sharks.
Exactly. And recent DNA work, similar to the Russian study but on a broader scale, showed that some of the mushrooms we were calling Agrocybe were not closely related to the core Agrocybe group at all. They belonged to a completely different lineage.
So who got evicted from the Agrocybe house?
The most famous evictee was the poplar fieldcap, Agrocybe agerita. This is a very popular edible mushroom, often cultivated in Europe and Asia. It grows on wood, specifically on poplar trees.
And it's not an Agrocybe anymore.
Nope.
Yeah.
It has been moved to the new genus Cyclocybe, so it is now correctly known as Cyclocybe agerita.
Cyclocybe, it sounds dynamic.
Yeah.
How would I tell a cyclocybe from a true Agrocybe like our Pediades?
Well, the Cyclocybe species usually have a much more substantial, tough, membranous ring on the stem, a real ring, not the vanishing act we see in Pediades.
And under the microscope.
Microscopically, their spores lack that distinct germ pore we talked about earlier.
So no escape hatch on the spores.
Or at least a very different kind of mechanism. But the good news for us today is that our mushroom, Agrocybe Pediades, is the type species of the true Agrocybe clade. It got to stay in the house. It's the original fieldcap.
That's a relief. I hate having to learn new names if I don't have to. But it's good to know that the genus is getting cleaned up. It makes the identification of Pediades even more secure, because we aren't trying to force it into a box with a bunch of unrelated cousins.
Exactly. Clarity is the ultimate goal of taxonomy, even if the road to get there is a little messy.
Speaking of messy, let's talk about the messy business of eating things you find on your lawn. We've established that Agrocybe Pediades is, in some sources, technically considered edible.
Technically edible. Those might be the two most dangerous words in all of foraging.
Right. It feels like it's in the same category as, cardboard. You can eat it, but why on earth would you want to?
The culinary value is virtually zero. It's tiny, the flesh is thin and watery, and remember that smell we talked about?
The raw flour. Farinaceous.
That translates directly to the taste. It's mealy and bland. You would need to pick 100 of them just to make a tiny garnish, and the texture would probably be pretty disappointing.
But the real reason to avoid it isn't just that it tastes like wet dough. It's the company it keeps. It's the neighbors.
It's the LBM danger zone, and I cannot stress this enough. When you are picking small brown mushrooms from a lawn, you are walking through a minefield.
Okay, so let's map out that minefield for everyone. Who is the biggest threat? The number one mushroom you do not want to confuse this with.
The biggest threat, the one that will absolutely kill you, is Galerina marginata. It has a common name. The funeral bell.
Great name. Terrible, terrible fungus.
It contains amatoxins. These are the exact same toxins found in the death cap Amanita phalloides.
And what do they do to you?
They are cytotoxic. They stop your cells from making proteins. Specifically, they inhibit an enzyme called RNA polymerase II. Your liver basically just melts. It is a slow, agonizing death that can take days to finalize, often requiring a liver transplant to even have a chance of survival.
And Galerina looks like Agrocybe.
To the untrained eye, absolutely. They are both small, they are both brown, and they can both appear to have a ring or remnants of one on the stem.
But what about habitat? Isn't there a difference there?
Usually, yes. Galerina typically grows on wood, logs, stumps, that sort of thing. And Agrocybe is usually in grass or dung.
But didn't we just spend 10 minutes talking about how much Agrocybe loves wood chips?
Well, we did. And Galerina also loves wood chips. And on top of that, sometimes there's buried wood in a lawn, old roots, construction debris. So the habitat overlap is very, very real.
So if you're holding one in your hand and you can't rely on habitat alone, how do you tell them apart?
The spore print. It is the one definitive test. Agrocybe is cigar brown. Galerina is rusty brown.
Cigar versus rust. I have to say, that is a terrifyingly subtle distinction to bet your life on.
It is. The lighting conditions matter. The color of the paper you print on matters. Your experience matters. If you make a mistake, you die. That is why the rule is, don't eat little brown mushrooms from the lawn, period.
Are there other threats in that minefield?
Oh, yes. In the exact same lawn, you will almost certainly find Peneolus phanicicii, the lawnmower mushroom. It's probably the single most common lawn mushroom in North America.
Is that one deadly?
No, not deadly. It's complicated. It's considered mildly toxic, usually just causing some gastrointestinal upset. In some cases, it can be weakly psychoactive, but it's not a reliable edible or recreational. It mostly just makes you feel sick.
And how does it look different from Agrocybe?
The gills. The gills are the giveaway. Peneolus gills look mottled. They look like they have black and gray patches all over them.
Why is that?
It's because the spores don't all ripen at the same time. So you have patches of mature black spores right next to patches of immature, paler, sterile cells. It gives the gills a speckled, uneven look. Agrocybe gills, on the other hand, are a very uniform brown.
And the spore print?
The spore print of Peneolus is jet black or a very dark purplish black, not brown at all.
Okay, what about the flip side? What about the one good edible that grows in grass? The fairy ring mushroom.
Marasmes oreades. A choice edible. It's delicious, nutty. It grows in rings in the grass, just like Agrocybe sometimes can.
So how do you tell that one apart?
Two key things. First, Marasmes has a white spore print, completely different color family. And second, its gills are distant. They are spaced very far apart. You can see a lot of space between each gill. Agrocybe gills are crowded and close together.
So just to recap the neighborhood, in 1 square foot of lawn, you could have the boring Mealy Agrocybe, you could have the delicious Marasmes, you could have the tummy ache-inducing Peneolus, and you could have the liver-destroying Galerina.
All potentially growing within inches of each other. It's a high-stakes game of one of these things is not like the other. And frankly, for a mealy-tasting mushroom, the risk-to-reward ratio is just nonsensical.
I agree completely. But you know who has absolutely no concept of risk-to-reward ratios?
Your dog.
My dog.
Yes, the canine factor. This is a very serious topic for anyone who has pets.
The toxicology reports from NEMA, the North American Mycological Association, they make it clear that dogs are frequent victims of mushroom poisoning. Why is that?
Well, dogs are scavengers, first of all. But more specifically, their incredible sense of smell leads them into trouble. Some mushrooms produce amine-like odors.
Amines. Like in fish.
Exactly. To us, it might smell foul or fishy, but to a dog, it can smell like rotting meat or a source of protein. It's irresistible to them.
Does our Agrocybe smell like that?
Not really. As we said, it's mealy. But it grows in mixed stands with mushrooms that do. For example, many species of Inocybe, they often grow in the exact same mulch or grass. And some Inocybes have a very distinct spermatic or fishy odor. Dogs love it. They just inhale them.
And if the dog eats the Inocybe along with the Agrocybe, what happens?
Inocybe species contain Muscarine. This is a fast-acting neurotoxin. It affects the parasympathetic nervous system.
And the symptoms have a really horrible acronym, don't they?
They do. SLEDGE.
Okay, let's break down SLEDGE for everyone.
It stands for salivation, lacrimation, which is tearing eyes, urination, defecation, gastrointestinal distress, and emesis, which is vomiting.
So basically, every liquid in the dog's body tries to leave at once.
That's a very graphic but accurate description. It is a full-body fluid expulsion. It can also cause bradycardia, a dangerously slow heart rate and constriction of the pupils.
That sounds absolutely horrific.
It is. The good news is, for pure muscarine poisoning, there is an antidote, a drug called atropine. It reverses the effects of muscarine very effectively if administered in time.
Okay, so if the dog is drooling and vomiting, the vet gives atropine. simple enough.
Ideally, yes.
Yeah.
But here is the catch. This is where the complexity of the fungal poison landscape becomes so dangerous. What if the dog didn't eat an Inocybe? What if it ate an Amanita muscaria or an Amanita pantherinoides that was growing nearby?
Those are the fly agarics, the classic red ones with white spots, or the yellow or tan ones. They contain completely different toxins, right?
Completely different. They contain ibotenic acid and mucimol. These are isoxazole derivatives. They don't cause SLA-DGE. They cause neurological symptoms. Confusion, ataxia, stumbling around like they're drunk, and very often a deep coma-like sleep.
So the dog just looks like it's in a coma.
Yes. Now, here is the critical veterinary dilemma. If a veterinarian sees a comatose dog and suspects mushroom poisoning in general, but they don't know which mushroom it was, and they administer atropine.
Thinking it might be the SLA-DGE-inducing muscarine.
Exactly. If they give atropine to a dog that is actually suffering from ibotenic acid toxicity, it can be disastrous. The source material is very clear that atropine can exacerbate the effects of ibotenic acid. It can turn a situation the dog would have recovered from into a fatality.
That is a terrifying interaction.
Yeah.
So the treatment for one type of mushroom poisoning is a poison multiplier for another.
Correct. For ibotenic acid poisoning, the Amanita sleep, the recommended treatment is often just supportive care, 4D fluids, monitoring their breathing, and just waiting. The dog usually wakes up on its own in 6 to 12 hours, sometimes a bit groggy, but otherwise fine.
This just emphasizes why identification is so incredibly crucial. If you just rush to the vet and say, my dog ate a mushroom, you aren't giving them enough information to make the right call.
You have to bring a sample, even if it's half chewed and covered in vomit. Put it in a paper bag, never plastic, because plastic just speeds up decomposition and turns it into slime, and bring it to the vet. Or take good photos and post them to a verified identification group on social media where real experts hang out. Knowing Agrocybe versus Inocybe versus Amanita changes the entire medical protocol.
So our mushroom, Agrocybe Pediades, isn't the dog killer itself, but it's the innocent bystander in a very, very dangerous crowd.
It's the guilty by association mushroom.
Wow. We've covered a lot of ground here. I mean, we've gone from a microscopic germ port to the global agricultural supply chain, from Russian genetics to Brazilian cattle dung. When you step back and look at Agrocybe Pediades now, after all this, how has your view of it changed?
I think I've stopped seeing it as just noise. At the very beginning, we called it background noise. Now I really see it as a signal.
Of what?
A signal of connection. This is a species that has fundamentally tied its fate to ours. It's the everywhere mushroom, because we are everywhere. It demonstrates that human activity doesn't just destroy nature, it reshapes it in bizarre ways. We are spreading these species all around the globe, creating a kind of homogenized planetary lawn.
That's a powerful image, the planetary lawn.
Mm-hmm.
And Agrocybe Pediades is like the grass growing in the cracks of the sidewalk of the world.
And it forces us to question what a species even is. If a mushroom in Russia and a mushroom in Brazil are genetically the same thing because of human activity, are we freezing its evolution, or are we speeding it up by introducing it to new environments? The existence of that little var, Spores, suggests that nature is still trying to do its own thing, drifting away where it can.
It's also just a profound reminder of the complexity hidden in the mundane. We walk past these things every day thinking they're simple. Oh, it's just a little brown mushroom. But inside that little cap, there's this incredible machinery. The vanishing veil, the Hygrophanous color change, the genetic library that stretches back millions of years.
And the chemical warfare defenses that its neighbors are deploying all around it. It's a silent battleground and a silent, thriving civilization right there under our boots.
So I think here is the takeaway for you, the listener. The next time you are out walking your dog or cutting through the park or even just weeding your garden, don't just glaze over the little brown mushrooms.
Stop for a second, crouch down, take a look at the cap. Is it cracking? Is it sticky from the morning dew? Look at the gills underneath. Are they that specific, dull cigar brown?
Try to catch the veil before it vanishes. It's a true blink-and-you-miss-it moment of biology.
But respect the danger. Absolutely respect the Galerina and the Inocybe that might be standing right next to it. Observe, photograph, identify, but please do not consume.
The world is full of these invisible neighbors. Agrocybe Pediades is just one of them, but now you know its name and you know its story.
And that makes any walk a lot more interesting.
Thanks for diving deep with us. Keep looking down.
Stay curious.